Howdy, my name is Hayden Elliott, and today I will be presenting Apis mellifera propolis, a natural approach to eliminating Streptococcus mutans levels. To start with, we need to talk about the bug of the hour, Apis mellifera, or more commonly, the western honeybee. The western honeybee, Apis mellifera, is one of the most widely distributed and economically important bee species globally. There are numerous subspecies of Apis mellifera adapted to different climates and environments, reflecting the bee's complex evolutionary history and its interactions with human societies over millennia. Through its quite extensive history, humans have found unique ways to use the bees and their products. Some common products are honey, wax, and propolis. Today, we will cover propolis and its unique abilities and uses for modern medicine. Propolis. Propolis is a resinous product produced by honeybees. For honeybees, it acts as a nest support tool and antimicrobial for bee health. It has long been known as a household remedy and cure-all. However, scientists have since started to study and evaluate its effectiveness at fighting microbes within the human body. One such microbe is Streptococcus mutans. This is a gram-positive bacteria that is commonly found in the mouth. This bacteria is one of the primary causes of tooth decay in the world. Largely, this bacteria feeds on sugars such as fructose, glucose, and sucrose. For this reason, it is important to recognize the importance of this bacteria and the different ways we can fight it. This brings me to my research question. Can Apis mellifera propolis stop the production of Streptococcus mutans? First up, we have a study by Ku et al. Researching the effects of new varieties of Apis mellifera propolis on Streptococcus mutans. In this study, Ku and team found that ethanol-derived concentrations of propolis significantly reduced the amount of mutants present in cultures. Reading this graph, we see that on the left-hand side, we have the size of the mutants colony. Here, the larger the number, the more bacteria present. We want to reduce this number. On the bottom is the concentration of the ethanol-extracted propolis, which I will refer to as EEP. Looking at this graph, we see that as the concentration of EEP increases, the overall size of the bacterial colony decreases. This shows a strong correlation between the antimicrobial properties of propolis and its effects on decreasing Streptococcus mutans levels. It is important to note here that the three different curves that are charted represents different strains of honeybee propolis. Propolis being derived from plants changes based on the local flora present, and this is largely dependent on the geographic location. However, for this presentation, we are going to look at the effect of propolis on bacterial concentrations, and the geographic differences can be left for future projects. Here we have a similar graph as before that is used to corroborate Ku and team's claims of the inhibitory effects of propolis on mutants levels. This graph is to further illustrate the legitimacy of propolis as a way to manage mutants levels. Moving on, we have a different study by Ku et al. Looking at particular flavonoids versus oral bacteria. This graph compares a control curve of uninhibited bacterial growth versus inhibited bacterial growth. It is important to point out that the flavonoids are considered the active ingredient in propolis that has been found to produce its antimicrobial properties. Furthermore, here we see that there are a few different strands of bacteria tested. These strands are mainly to show the inherent effect that farnesyl, the flavonoid in question, has on bacteria. These strands are other commonly found oral bacteria that is found in the mouth. Moving on, we have another study by Hegde et al. Looking at the effects of propolis on mutants levels during an in vivo experiment. This study examined children's saliva before and after application of propolis. The results when compared to the control group show a pretty stark contrast in the mutants' growth when paired with propolis. Overall, this study is very important in the continuation of propolis research as it shows the effects of propolis on Streptococcus mutans within an organism. The larger implications of this study show that propolis has a legitimate claim to fame in inhibiting the growth of oral bacteria as a method of maintaining oral health. But there's still much more research needed to be done. Overall, we can see that propolis is a legitimate natural way to fight cavities. We saw that it has antimicrobial properties that significantly reduce levels of bacterial growth across not only Streptococcus mutans, but other strains as well. Lastly, we saw that propolis does work as an oral application on real people. Lastly, why is all this important? Well, cavities are a legitimate concern for everyone who has teeth, and finding new ways to fight them can only benefit the global oral health. By exploring new natural avenues, we as scientists can find ways to work with the environment to fight our problems. Propolis is not only a way to fight cavities, it is a way to bridge the gap between the environment and our needs as a society. Through efforts like exploring propolis, we uplift and raise awareness for how awesome the environment is and how we can use it to our advantage. Studies like the ones I used here show a great encouragement for bee-friendly agriculture and can help uplift local apiaries and find new ways to market bee products. Lastly, I just want to say thank you for listening, and here are some references I used if you want to continue reading. Thank you, and have a good day.